students, may Almighty's peace be upon you. I'm very happy to meet you again in the last session of this lesson 1, Asexual and Sexual Reproduction in Plants. So we were discussing so many things in this first lesson, and even then you know very well that all the parts are very important. We discussed how the gametophytes are formed, gametes are formed, then fertilization, post-fertilization, and embryo development. Now in this session, we will be discussing about the structure of seeds. So as we know earlier, the seeds with two cotyledons are called as dicotyledons or dicots. Same way, the seeds with one cotyledon is called as monocotyledon or monocots. So we were discussing in our endosperm topic that all the monocots, maximum of the monocots will have endosperm in them, which gives nutrition to the developing embryo, isn't it? So monocots, endosperm is the major part in monocot seeds. At the same time, in dicot seeds, we do not find such thing. Okay, only two cotyledons. These cotyledons will give nourishment. So in today's topic, we will be discussing about structure of monocot seed and dicot seed, and how the fertilization occurs without involving the union of gametes. We will be discussing. Then polyembryony, more than one embryo is formed, polyembryony, and Parthenocarpy. So these are the topics we will be discussing in today's session. Now let's discuss about the structure of dicot seed. Speak about dicot seed. Dicot seed, structure of dicot seed. So we know the major structure, the outline structure is given here. You can see two cotyledons, that is why it is dicotyledon. This it's a overall seed actually. So dicot seed. Now let's discuss the parts present in dicot seed. Okay, the first one is the stalk of the seed. So seeds are attached to the fruit wall by a stalk. The seeds are attached to the fruit wall by a stalk called funiculus. As we discussed the same in LS of ovule structure, we discussed funiculus. Same way, the stalk of the seed coat, which is attached with the fruit wall, is called as funiculus. Point number 1. Then this funiculus will disappear and there will be a scar in the seed. You can see. So the funicle, the stalk disappears, degenerates, and you will have a scar there. So that scar is called as hilum. Okay, when it disappears, totally when it disappears, it leaves a scar called hilum. Then third one, below this hilum, there will be an opening called as micropyle. We know it already in our LS of ovule. There is an opening at the top of the ovule. Same way here also, micropyle is the opening. The purpose of this micropyle is the water and oxygen enters the seed through this micropyle only. So micropyle is the opening below the hilum. This is the third part. And now coat, seed covering. Okay, seed covering, outer coat, the outer coat that is developed from integuments of the LS of ovule. So the outer coat is testa. And we have discussed already in the post-fertilization changes, the inner coat, that is the inner integument becomes the inner coat, which is called as tegmen. Now two cotyledons are laterally arranged on the embryonic axis. This is the embryonic axis, and two cotyledons are present laterally. Okay, this cotyledons only will have nourishment. In case of monocot seeds, endosperms will have nourishment. It gives nutrition to the embryo. But in case of dicot seeds, these cotyledons will have nutrition. It will supply nutrition to the embryo. So two cotyledons are present. And if you see these two cotyledons, and a portion of this embryonic axis is projected above and below. You can see somewhat projected above and below. Okay, the portion which is projected above is called as plumule. Some pronounce as plumule also. It's plumule. So plumule. That is the portion which is projected above, plumule. This plumule only will develop into shoot system. Further, it will develop into shoot system, and which is below. Okay, that is called as radicle. Radicle, which will be formed as root system. So in dicotyledons we have shoot system and root system. The primary portion is plumule and radicle. Okay, you have plumule and radicle here. Then the above the level, so you can see this is the level of the cotyledon. Above the level of the cotyledon, the portion above the level of the cotyledon is called as epicotyl. Epicotyl, the above the level of the cotyledon, the portion is called as epicotyl, and the portion below the level of the cotyledon is called as hypocotyl. Hypocotyl. This is the structure of dicot seed. Okay, come on, let's recall once again. First is funicle. Funicle is nothing but the start of the seed wall, seed which is attached to the fruit wall. First, funicle. Funicle is second one. It disappears and forms a scar called as hilum. Point number 3. Below the hilum there will be an opening through which water and oxygen enter into seed. That is micropyle. Micropyle. Then fourth, seed coat. Outer seed coat is called as testa, and inner seed coat is called as tegmen. Then next one we have the embryonic axis. Two cotyledons are present in the embryonic axis, and the embryonic axis projects outside, which is projected above is called as plumule, which will be developed into shoot system, and the portion below the embryonic axis, so this is called as radicle, which will be developed into root system, and the portion of the embryonic axis above the cotyledon is called as epicotyl, and the portion below the level of the cotyledon is called as hypocotyl. Now we should learn about monocot seed. Now when we discuss about monocot seed, we can give example as paddy. So the whole seed of the paddy is called as caryopsis. You know the structure of a rice paddy, isn't it? So there will be a brown color outer covering. Totally, that is the only thing we call as totally. It is a seed actually. So now let's discuss about the internal structure. What are the things present inside the monocot seed? So very simple. As we all know, here only one cotyledon is present. And as we discussed earlier, the monocot seed totally, the maximum of the monocot seed will have endosperm. So the total, this region fully, it will have endosperm only. This is endosperm. So see, this region is endosperm. So the maximum of the monocot seed will be endosperm, which gives nutrition to the developing embryo. So this is endosperm, and endosperm will have a definite layer here. A layer is present here which separates the seed from the endosperm. Okay, other contents from endosperm by epithelium. By epithelium. Okay, the layer is called as epithelium, and this seed will have this embryo set, totally this embryo, it will have a shield shaped one cotyledon. This is the shield shaped one cotyledon. Only one cotyledon is present. In case of dicot seed, we discussed that there are two cotyledons were there, but here only one cotyledon is present, which is called as scutellum. Scutellum. So this is called as scutellum. Scutellum. This scutellum, this monocot, this cotyledon will have the portion for plumule and radicle. So plumule and radicle. So we have already discussed what is the role of plumule and what is the role of radicle. Plumule develops into shoot system and radicle develops into root system. So it will have plumule and radicle. But the thing is, this plumule and radicle are protected by some covering. A protective sheet will be present for this plumule and radicle. Okay, so plumule, this is plumule, and there will be a protective covering. You can see a protective covering is present, isn't it? So this protective covering, a layer, the plumule is surrounded by a protective sheet called as coleoptile. Coleoptile. Okay, the plumule is surrounded by a protective sheet called as coleoptile. And what for radicle then? It is coleorhiza. Coleorhiza. So coleoptile and coleorhiza. To avoid confusion, which is coleoptile and which is coleorhiza, I remember like plumule P, so it is coleoptile. Radicle R, so coleorhiza. Okay, so need not to get confusion with this coleoptile and coleorhiza. So these are the major parts in monocot seed. Okay, I'll just recall. See, so this is the seed coat and fruit wall, and the seed wall is made up of aleuron layer, and the major portion of the monocot seed constitutes of endosperm, which gives nutrition to the developing embryo, and it will have a one shield shaped cotyledon called as scutellum. And this endosperm and scutellum is developed by a layer of, that is, there will be a distinct layer of epithelium. In this scutellum we have plumule and radicle, which develops into shoot system and root system. But this plumule and radicle are protected by covering called as coleoptile and coleorhiza. See, coleoptile and coleorhiza. This is the structure of monocot seed. The next new topic to learn is apomixis. Apomixis, it's a new terminology actually. So actually we have amphimixis. Amphimixis is by fertilization. A union of male and female gametes leading to fertilization is called as amphimixis. We know that earlier. But now the term apomixis was coined by Winkler in 1908. Winkler in 1908 coined apomixis, that is, there is no fusion of male and female gametes. No fusion. So, by that, if embryo is developed. So now we have a confusion. Will it be developed like that? Yes, of course. There are some plants which will develop like that without seed. Okay, the seed will not be fertilized. Okay, so that is said to be apomixis. That is reproduction without involving the gametes. No fusion of male and female gametes is called as apomixis. So we can say that our Indian scientist P. Maheshwari, he told that it is a substitution for sexual system. Substitution without involving the gametes. Some methods are there for reproduction. Okay, so he classified into this apomixis into recurrent and non-recurrent. Recurrent and non-recurrent. Recurrent apomixis means generally meiosis will not occur. Maximum meiosis will not occur, and the new embryos will form. Okay, the meiosis, the divisions will not occur. Non-recurrent apomixis means after meiosis, then the embryo sac will develop into seed, but there is no fertilization. So non-recurrent apomixis. Now let's discuss about recurrent apomixis. So recurrent apomixis, there are two types. One is vegetative reproduction and agamospermy. Vegetative reproduction we have discussed in the first part of the lesson. So in the artificial system, natural we know. So in vegetative reproduction we discussed bulbs, that is onion, allium cepa. Okay, so it develops without seed, even the bulb of the onion develops into new plant. Okay, that is there. Sucker, runner, bulbils, all they are the examples of vegetative reproduction. Without the fertilization, the new plant arises. Okay, now agamospermy is nothing but the embryos are formed. Embryos are formed by eliminating the meiosis. There is no meiosis. Generally, no meiosis will occur. That is agamospermy. And no syngamy also, no fusion of male and female gametes occurring here. So there are three types of agamospermy: adventive embryo, diplospory and apospory. Adventive embryo, diplospory and apospory. Let's see adventive embryo. Adventive embryo means the diploid sporophytic cells. After diploid sporophytic cells, from nucellus, the embryo cells are formed. The new embryo will formed from nucellus. So that is adventive embryo. It takes place in citrus and mandifera, our mangrove. So this is also called as sporophytic budding. Sporophytic budding. Okay, adventive embryo. Diplospory. Here in diplospory, a diploid embryo. Okay. The megaspore mother cells, the female gametophyte, the megaspore mother cells, without regular meiosis, it develops into new plant. Without regular meiosis, okay, no meiosis. That is diplospory. Apospory, a small change from diplospory. That is, you don't have the megaspore mother cells will form 4 cells, but after that, it will disappear. Okay, it will disappear. So that is called as apospory, and that will develop into a new plant. That is apospory. So apomixis is nothing but generally without the fusion of male and female gametes, we have certain plants. That is what we discussed here. The next topic is polyembryony. From the name itself we can understand. Poly means many. Embryony, embryos. So generally one embryo will be formed in the embryo sac, but if more than one embryo is formed, it is called as polyembryony. That also happens in plants. Okay, so that is polyembryony. First it was discovered by this polyembryony. We will be wondering to know, Anton van Leeuwenhoek, the one who found out the microscope, found polyembryony. In 1719 itself, he observed polyembryony because more than one embryos are formed here. So based upon the text, we need not to discuss much about polyembryony, even then a little knowledge. Cleavage polyembryony, when the embryos cleave. Okay, so that is cleavage embryony, polyembryony. So orchids are the best example for cleavage polyembryony. So the embryo cleaves. The second one is formation of embryo by cells of the embryo sac other than egg. Other than egg means where it will be. We know that in the embryo sac we have egg. Other than that, what do we have? We have, yes, antipodals we have, synergids we have, isn't it? So those things, so other than egg, synergids or antipodals or endosperm, if they form embryo, that is formation of embryo by cells of the embryo sac other than egg. Okay, so we have a few examples in synergies, some acistolochia and an endosperm banalothoria, balanophoria, sorry, an endosperm from endosperm. So there are many examples. So other than egg, antipodal synergids all form as new embryo. Then third one is development of more than one embryo sac within the same ovule. In same ovule, embryo sac, more than one embryo sac is formed. Okay, so that is general. In Casuarina, it occurs. Finally, activation of sporophytic cells. Activation of sporophytic cells means new cell as interhumans from there. Okay, these sporophytic cells, if they develop, then it is fourth type, activation of sporophytic cells. In citrus, we can see such type. Why do we go for this? Why? What is the use of this polyembryony? One thing to say, they are found virus free. They are found virus free. That is the major advantage of this polyembryony. The last topic of the lesson is Parthenocarpy. You would have learned, you would have heard it, because even in zoology we have discussed parthenogenesis, isn't it? Without the act of fertilization. Same way, Parthenocarpy is nothing but without the act of fertilization, some fruits are developed. So that is Parthenocarpy. And such fruits are called as parthenocarpic fruits. But the thing is, all the parthenocarpic fruits are seedless fruits. They are generally seedless fruits. Now you tell me, what will be the advantages of this parthenocarpic fruits? Now you just see, when you go for purchasing fruits, you have grapes with seeds and without seeds. Seedless grapes. So which one do you prefer? Obviously we go for seedless, because we need not to split the seeds all, isn't it? So that's the thing. So when we see the significance, what is the importance of this parthenocarpic fruits? One thing, it is used in horticulture. In horticulture this is used. Second, commercial importance is there. The purchasing, they'll purchase more for seedless fruits. Commercial importance is there for this parthenocarpic fruits. Third one, in the preparation of jams, jellies, pickles, they go for seedless fruits. Can you say why? Why do they go for seedless fruits? Because removing the seeds will be a process if they have seeds. So in these cases, in parthenocarpic fruits, such problem does not occur. So in the preparation of jams, jellies, pickles, all we use parthenocarpic fruits. Finally, the edible portion is more since there are no seeds. So high edible portion we can say. So these are some of the importances of horticulture. My dear students, by this we have completed first lesson, asexual and sexual reproduction in plants. So we covered so many topics. Asexual reproduction. In asexual reproduction we discussed vegetative reproduction and many modern methods, conventional methods. Then we discussed about flower morphology, reproductive parts of the flower, androecium and gynoecium, about the microspore and megaspore formation, how anther pollen grains are formed, structure of anther, structure of pollen grain, LS of ovule, the female reproductive part, how the pollen grain enters the ovary through the style. We have discussed so many things, and interestingly about pollination also, and some extra topics like Apomixis, Parthenocarpy, Polyembryony, etc. So now it's time for you, it's your duty to learn this first lesson. Thank you all.